Okay, today we are going to go over case studies 24 and 46, starting with case 24, which is interferon gamma receptor deficiency. Question number 1. Macrophages secrete what after phagocytosing pathogens, and what does this cytokine do? So it's interleukin 12, and what interleukin 12 does is excuse me, it triggers the maturation of Th1 cells and also suppresses the maturation of Th2 cells, and also induces the production of interferon gamma in T cells and natural killer cells. And the most important cytokine involved in macrophage activation is interferon gamma, and it activates macrophages. When they bind interferon gamma, there's the induction of there's a transduction pathway that's activated. It's a JAK-STAT pathway, and this allows macrophages causes them to form phagolysosomes, so they can engulf pathogens previous to this, but it's upon this activation process that they're able to actually digest the pathogens that they've taken up, that they've phagocytosed. And then also this triggers an autocrine signaling response where the macrophages express TNF receptors, and they also secrete TNF alpha, which causes them to produce antimicrobial compounds. And then in addition to that, when MACs are activated, they present CD40, B7, and MHC class 2 for activation of CD4 interactions with CD4 cells. Would you expect an inactivating mutation of STAT1 to have similar or different phenotypes compared to Clarissa? What about an activating mutation? Inactivation of STAT1 would have a similar effect, so it would be a similar phenotype to what is seen in the case study, and that's because STAT1 is part of the interferon gamma response. STAT1 is needed for it's the transcription factor that actually, once activated, enters the nucleus and affects the activation of the genes in the macrophage. So it would be a similar phenotype if there was a STAT1 inactivation mutation. If it was an activating mutation, that might cause macrophage activation without external stimulus, which could increase inflammation. Mycobacterial infections are prevalent in patients with AIDS. Why is this? Why can't these patients' MACs fight mycobacterial infections effectively? And that is because in patients with AIDS, they don't have functional CD4 T cells, so T cells are not activating macrophages, which makes these individuals susceptible to mycobacterial infections. And so, since macrophages aren't being activated, then the macrophages can't contain or clear the infection. Number 5, Clarissa did not develop granulomas as a result of mycobacterium infection and had mycobacteria in her blood. Explain why interferon gamma receptor deficiency would cause these phenotypes. It's what we've already been kind of talking about here, or me, I guess, what I've been talking about here, where Without a functional interferon gamma receptor, then macrophages don't respond to the T helper cell signaling that would activate them. And so since the MACs are not activated, they are not producing granulomas, which help to contain the infection. And so the lack of granulomas means that the mycobacterium can spread to the blood, and it's because of the interferon gamma receptor deficiency, the breakdown in the activation of that JAK-STAT response that is causing this phenotype. Number 6, do you predict that Clarissa would have a problem fighting off viral infections? Why or why not? Yes. Without proper macrophage activation, they would not participate in antigen presentation, since macrophages are APCs, and so they process and present viral protein epitopes. Okay, moving on to case 46, lepromatous leprosy. What cytokines do Th1 and Th2 cells produce? Th1 cells produce IL-2, interferon gamma, leukotrienes. Th2 cells produce IL-4, IL-5, IL-10, and IL-13. What cytokines favor Th1 cell production and also Th2 cell production? So Th1 cell production is favored by interleukin 12 and also interferon gamma, and then Th2 cell production is favored by IL-4. What is the difference between tuberculoid and lepromatous leprosy? There is I think it's figure 46.4. That is a good reference for this, but basically tuberculoid, the tuberculoid version of this infection is a Th1 response. So, and it's characterized with low bacteria levels. It's not as infectious as the other version. And then also you see the formation of granulomas. There's local inflammation. Macrophages are being activated, and so they are involved in engulfing and digesting these bacteria, and also forming these granulomas that help to keep the infection from spreading. And then also the immunoglobulin levels are not elevated because this is a Th1 response, not a Th2 response, where in contrast the other version is a Th2 response, which, since macrophages aren't being activated, we see them engulfing the bacteria, but there is actual growth taking place inside the macrophages, since there isn't that formation of the phagolysosome and the digesting of this pathogen. The infection is not localized, there's no granuloma formation, there's no containment, so it spreads. And also, since this is a Th2 response, which is an antibody response, there's elevated immunoglobulin levels. List some cytokines that could be given to patients to treat lepromatous leprosy. How would they specifically work to cure or lessen the severity of the disease? So IL-12 and interferon gamma would stimulate a Th1 response and macrophage activation. So this would basically trigger a response that would be the tuberculoid form, which would be not as infectious. It would be contained, localized, and macrophages could clear the infection. And then also, another thing that could be considered would be the blocking of other cytokines such as IL-4, since IL-4 is involved in the stimulation or production of those T helper 2 cells, which are detrimental to the patient. Ursula is prone to asthma and severe allergies. Why is this the case? She is stimulating a Th2 or antibody response, and having increased antibody levels is going to make her more prone to autoimmune diseases such as asthma and allergies. And that's it.